Good evening. So for those of you in the back, just to let you know, there are some seats up front. Do not be shy. Come and fill them in. You're all welcome. For those of you who don't know me, I'm Tom Sharkin, an assistant professor in the departments of microbiology and statistics here at Oregon State University. My lab focuses on the contribution of the gut microbiome to human health. So you can imagine I'm on pins and needles about this evening's lecture. I also have the distinct pleasure of welcoming you all here tonight to this particular event. This event is being hosted by the newly established Oregon State University Microbiome Initiative, which we refer to as OMBI, that's a play word. OMBI is a research and training and outreach program here at this university that applies an interdisciplinary approach with a focus on microbiomes and what they're doing, how they do it, and their social transformative applications. And tonight's lecture is particularly exciting because this represents the public inauguration of this event at the university. And we hope to have similar types of lectures here in the future and look forward to your engagement in those events. Now, I have to say one really important note at the very outset of all of this is that we would not be here tonight in the same room if it weren't for the generous support of the departments of microbiology, the College of Science, the College of Agricultural Science, and the OSU Research Office. We owe them a tremendous debt of gratitude. But to kick things off, I'd like to welcome to the podium tonight Sastry Pantula, the Dean of the College of Science, who's been a true champion of this initiative and microbiome research in general, to come say a few words.
Thanks, Tom. Again, there are more seats in the front here. Please do come on up after you get your food. What a crowd, really. Wow, it's impressive. I really didn't know that there's such a fan club for microbiome. Or it must be our distinguished speaker who can say it's not exactly rocket science. The launch of OSU Microbiome Initiative, OMBI, this spring represents years of hard work and collaboration across departments, colleges, units, and disciplines. As you all know, collaboration is a hallmark of OSU, which makes our campus a good home and an incubator for the big ideas. Today, there is an urgency driving microbiome research globally. With chronic diseases on the rise, ever-increasing ecological disruptions, and a growing need to improve agricultural productivity, scientists are looking to microbiomes for answers. The tiny things, the world of seeing the unseen, these microscopic organisms may hold the key to improving animal, plant, human, and ecological health. Again, the term microbiome has worked its way into our national dialogue. As a result, support and funding for microbiome research has grown substantially. Between 2012 to 2014, annual federal investment into microbiome research tripled, with more than $922 million invested. In early 2016, when the White House Office of Science and Technology Policy issued a national call for new microbiome research, Tom Sharkin galvanized OSU researchers to develop a collaborative proposal that has resulted in OMBI, our OSU Microbiome Initiative. As a nationally recognized microbiome expert, Tom brings an expertise in both microbiology and statistics that gives him a holistic and unique perspective on microbiomes. And I want to take a moment to recognize the incredible leadership Tom has provided for this important microbiome initiative on our campus, regionally and nationally. Let's thank Tom. So OMBI will help attract sustained external funding to OSU faculty who have already received $10 million in grants for microbiome studies in the last 21 months alone. I'm sure I saw Cindy in the background, back there, our Vice President for Research in the audience. She's thrilled about this, and we also appreciate her support and leadership for the research on our campus. OMBI reflects the values of our college, scientists collaborating and striving for excellence, harmony, and diversity. Through these partnerships, we will better understand how microbiomes operate, while also providing training in microbiome research methods, engage our students, the next generation of leaders in science, and engaging more underrepresented scientists. During the last 2 decades, microbiologists have rewritten the scientific script on health and disease, origins and evolution of life, and the connection between the microbial world and every other species on the planet. We are advancing from an antibacterial view of existence to an all-encompassing beneficial view of the nature of microbes. And now, Ed Yong, our speaker today, and his best-selling book have played a large role in this transition. I'm really honored to introduce to you today the award-winning science writer and journalist, Ed Yong. Given this large crowd, I think we are all of us are looking forward to hearing his talk based on his New York Times bestseller, I Contain Multitudes. The book chronicles an important development in biology today, the fascinating science of the microbiome. Ed is one of today's most prominent and influential science journalists. He's a writer for The Atlantic, the magazine's first staff science writer in its 159-year history. That is very good news to hear.
We turned 160 last week.
Thank you, Ed, for helping us develop scientifically literate public. I personally appreciate that you are a part of our science word spreading team. It is so important in these challenging times, especially nationally now. With advanced degrees in biology, Ed has written on topics ranging from microbes, science policy, paleontology, and more. His work has appeared in Nature, the New York Times, Scientific American, the New Yorker, Wired, the BBC, Slate, among many, many others. He was the author of the widely read blog, Not Exactly Rocket Science, hosted by National Geographic and widely considered the centerpiece of his science writing career. Ed has received multiple awards recognizing his knowledgeable and effective communication of scientific phenomena. He received the National Academy's Keck Science Communication Award from the National Academy of Sciences in 2010. So let's hear from Ed Yong, whose humor and writing reflects a deep understanding and wonder about science. Please join me in giving Ed Yong a warm welcome to our viewer nation.
Hi, Oregon. You might be able to tell from the accent that I am not from around these parts. But I've not come to you from London today. I've come from DC, where I've recently moved, because I heard how much you love immigrants. So, that's right, and science. You love science too. No more heckling in the crowd. Okay, so I'm going to start this talk by taking you not to neither London nor DC, but to a Dutch town called Delft in the year 1675, so about 350 years ago, when a man named... When a man named... Jesus Christ. Tech person, help. when a man named Anton van Leeuwenhoek was about to change the world. This man is not a scientist, he is not a scholar or an academic of any description. He is just a civic servant and a draper. But he does have two really important qualities for our story. He is insatiably curious about the world around him. And he is also the creator of what are at the time the world's most powerful microscopes. They look like this. They look kind of like a weird door hinge thing. And in this photo, unfortunately, it is presented upside down and back to front. But this weird-looking object is capable of magnifying things about much, much better than anything else around at the time. And Leeuwenhoek turns it onto everything else around at the time. Everything he can get his hands on, bits of animals and plants and bits of water that he collects from the lakes and canals of Delft, from a urn that happens to collect rain outside his house. And wherever he looks, whatever sample of water he looks at underneath through his microscope, he sees the same thing. He sees tiny little living things existing in extreme numbers. These are microbes, bacteria, and Leeuwenhoek has just become the first person in all of humanity's history to understand that these things exist, that they exist everywhere and they exist in almost untold numbers. And this is just a momentous point in the history of science, going from the point where we understand that there is this hidden diversity of life. Leeuwenhoek wrote about what he saw, and he sent letters about what he saw to the Royal Society. If any of you has anyone here read any of Leeuwenhoek's papers? Yes, a few people. If you haven't, I highly recommend them. They are kind of hilariously informal. Man's not a scientist, so they feel kind of like 17th century blog posts. You will learn a lot about the weather in Delft in the summer of 1675. Now, if you were the editor of a scientific journal today and some random person who you've never heard of before, who has no scientific credentials, sent you a paper saying they had just discovered this entire group of living things that no one else has seen before and that no one else has the capacity to yet see, what would you say to that person? Well, in a kind of extraordinary feat of scientific open-mindedness, the Royal Society actually did publish his papers, and they sought to have his observations independently verified. And when they were, they accepted him. And by the time Leeuwenhoek died in, I think, 17, he was one of the most famous members of the Royal Society, and certainly its most published member. When he died, he bequeathed the Royal Society this black lacquer cabinet with a couple of dozen of his amazing microscopes with specimens mounted on them. But that cabinet vanished under mysterious circumstances and has still to this date never been recovered, which was a big problem because Leeuwenhoek was very oddly secretive about how he went about crafting his microscope. So when he died and the cabinet disappeared, very few people could sort of duplicate his observations because no one knew how to make the same tools that he made. And when asked about why don't you just teach someone how to make your microscopes, he said this. He said, "Not one man in 1,000 is capable of such study because it needs much time and spending much money. And over and above all, most men are not curious to know. Nay, some even make no bones about saying, what does it matter whether we know this or not?" So here, 350 years ago, is the father of microbiology complaining about impact and funding. He has a very strange attitude to open science, and apparently he hasn't heard about women. Fortunately, everything is different now. So having made much progress since Leeuwenhoek, we're in a very different situation. But one thing that has definitely changed is that we see microbes, we understand that microbes exist, and we do see them in a slightly different light. We see them like as germs, as things that cause disease. Because after Leeuwenhoek, many scientists in very quick succession established that some of the most infamous diseases to afflict humanity throughout its history were the work of bacteria. So things like gonorrhea, syphilis, leprosy, tuberculosis, cholera, or the plague, all of the above. All of these things were the work of germs. And so for the longest time, microbes became known as germs, as villains, as things that we had to destroy in case they destroy us first. And we tried to eradicate them. To this date, if we spot microbes on surfaces, we take that as a sign of dirt and disease and decay. The presence of microbes on surfaces, I'm sorry to anyone who has a beard in the audience. I'm not judging you. I don't know why they are. The presence of microbes on surfaces fuels all these headlines about how we have clearly made a poor job of cleaning the world around us. They are signs of dirt. We want to remove them. And this is a very unfair and misleading point of view, because only a small minority of microbes are responsible for infectious diseases. The vast majority of them are either benign or beneficial to us. Each one of us houses, at latest estimates, around 39 trillion microbes in our bodies, roughly one for each of our own human cells. Some of you may have heard a slightly different 10-to-1 ratio. Turns out that is fake news. That is a made-up numerator glommed on top of a made-up denominator to give a made-up ratio. But so probably 1-to-1. Whatever the actual ratio, clearly there are a lot of them, and they live with us to no ill effects. And indeed, microbes are the dominant forms of life on the planet, and they have been around for the longest time. Usually when I do this bit, there's a much smaller stage, so I'm going to have to run. Okay, if we imagine the full length of this room to represent the history of life on Earth, so that the Earth was created 4.5 billion years ago in that corner, and that we are now here at this edge, then humans evolved here. The dinosaurs ruled from, I'm guessing, from about like here to here. And multicellular life, so all the things that you're familiar with and you can know and see, probably evolved around here. So for the rest of life's history, from the time it first appeared on Earth, probably about here, it was entirely microbial, microscopic. You wouldn't have been able to see it with your naked eyes, which would have been fine because eyes only evolved around here. So we, and by we I mean humans and the rest of the animal kingdom, evolved in a world that was already dominated by microbes. We are just the icing on this very deep microbial cake. And so we evolved to live with microbes, to exploit them, and to depend upon them. Each of us here as individuals are really ecosystems in our own right, entire worlds with large communities of microbes sharing our lives with us. These microbiomes exist not just in our bodies, but those of every other animal. This, for example, is a sponge. This is one of the simplest animals that exists. It's just a couple of layers of cells sandwiching a jelly-like filling. But this too is home to a thriving community of microbes. So thriving that sometimes if you look at a sponge under a microscope, it's hard to make out the animal cells for the microbes that cover it. Ants live in colonies that can number in their millions, but every ant is effectively a colony of millions in itself. A polar bear walking alone through the Arctic wilderness is in fact filled and surrounded by life. Bar-headed geese fly microbes over the tops of the Himalayas. Sperm whales dive them down into the deepest oceans. And when these people first set foot on the moon, they were also taking a giant step for microbe kind. These organisms are not just hitchhikers in our bodies. They're not just stowaways that happen to ride along with us to no effect. They also grant to their hosts incredible abilities, sometimes essential abilities. This adorable creature is the Hawaiian bobtail squid. If it was sitting in this room right now, it would fit very nicely in the palm of my hand, about the size of this clicker. This animal has two light organs in its undersides that are full of glowing bacteria, and the light that they produce shining below the squid matches the moonlight welling down from above it, which means that any predator looking up at the animal can't make out its silhouette. It has a kind of invisibility cloak rendered in bacterial light. This creature is paracatenula. It is a type of flatworm. It has the amazing ability to regenerate its body from virtually any kind of injury. Chop it in two, you'll get 2 flatworms. Chop it in 10, you will get 10. This is in fact how people who work with this animal breed it. That ability to regenerate depends on the symbionts that fill its body, those dark-colored microbes that you see taking up nearly its entire length. As long as some of those are around, it can regrow itself. This is an aphid, one of a massive group of some 85,000 sap-sucking insects. They can only subsist on a diet of plant sap because they have bacteria living inside their cells that provide them with nutrients that are missing from that sap. Without those bacteria acting as living dietary supplements, these insects would never be able to survive on this exceptionally common food source. These creatures take the concept of supplementation to an even greater degree. These are riftier tube worms. They live in deep sea hydrothermal vents. And they have microbes in their bodies that provide them with all the nutrients they need, to the extent that these worms have lost their mouths, their guts, their entire digestive systems. Effectively, they don't eat because they have microbes to provide them with all of their nutrition. These white lines coming out of a burst insect cocoon are parasitic worms that kill insect hosts by vomiting toxic microbes into their bodies. And this bird is the hoopoe. It paints its eggs in a paste that contains bacteria that produce antimicrobial chemicals. And those defensive chemicals stop the chicks inside the eggs from contracting diseases. So all of these benefits, regeneration, light, supplementation, anti-disease defense, all of these are the kinds of abilities that microbes can bestow on their hosts. We humans do not do this. We don't regenerate, we don't glow in the dark. If any of you do that, please see me later. But we also rely on our microbes for all kinds of things. They help to digest our food, they help to train and calibrate our immune systems, they help to sculpt and shape our organs. All corners of biology are touched by their influence. And I've been writing about this area of science for, well, pretty much as long as I've been a science writer, which is about 11 years now. And I find it incredibly fascinating because it gives us a completely different view, a different understanding of our cells and the way our biology works. You know, I was always that kid who went to zoos, and now if I go to a zoo, I understand that every creature and visitor at the zoo is a zoo in its own right. And I understand that if I don't understand the microbial component of all of these creatures, I don't really understand them or myself. Because when we look at the world through this microbial lens, even really familiar aspects of our lives take on this kind of weird and wondrous new air. So consider something like breastfeeding. Breastfeeding is obviously a way for a mother to nourish a baby. But it is also much more than that, because something like 10% of breast milk contains these sugars that babies cannot digest. Those sugars are there to nourish microbes in the baby's gut, and particularly strains like the one on that slide, B. infantis, that in turn, that have evolved the ability to digest those sugars with incredible efficiency. And when it does so, it releases nutrients that nourish the baby's gut, that seal up the lining of the gut, it reduces inflammation. And so a mother, by feeding the microbes inside her baby's gut using breast milk, is not just nourishing her infant, but infantis as well. She's sculpting this entire ecosystem inside her child. She's engineering an entire world. And that world, in turn, engineers us. We know that the microbes in our bodies help to shape and sculpt the development of our organs. All animals grow from a single fertilized egg into an adult body with trillions of cells. And that process of development doesn't unfold just under our own steam, according solely to the instructions encoded within our own genes. It happens in partnership and in conversation with the microbes that share our lives. And we can understand some of those conversations by looking at creatures that lack microbes of their own, like these. Not the one on the right, that one's loaded with microbes. The one on the left is a germ-free mouse. It was raised in one of these sterile plastic bubbles in which it lived in its entire life, disconnected from the microbial world. This is one of the few creatures in the planet that does not contain multitudes. It's like an empty silhouette. And because it doesn't have any microbes in its body, it has all kinds of physiological problems. Its bones don't develop correctly, its gut, its blood vessels. And you can see this in germ-free animals from lots of other different species, in zebrafish, in insects. Time and again, we see that animals that lack microbes don't develop in the right way. Even if creatures have a rich, thriving microbiome, their health could also suffer. Sometimes changes in that microbiome have been linked to ill health and to disease. And when it comes to human health, changes in the microbiome have been linked to a really very long list of conditions indeed. A conditions list so long that it almost invites parody. In these cases, it is not that a single microbe is causing the illness in the style of, say, an infectious disease like plague or cholera. It is more likely that the entire community of microbes has changed in a way that negatively affects the host. And this sort of community level change is called dysbiosis, a term that I will come back to later. But for now, I want to stress that most of these associations between microbiomes and the human microbiome in these conditions are correlative. They are based on studies that can't really tell us whether the change in the microbiome is responsible for the disease, is a consequence of disease, whether it's both or whether it's neither. But there are some types of experiments which can speak to causality, that can hint at a causal influence. Those germ-free mice that I showed you before, if you take rodents that have the mouse or rat equivalent of a disease like, say, type 2 diabetes or inflammatory bowel disease or heart disease or whatever, and you take their microbiome and transplant them into one of these germ-free rodents, sometimes you'll see that the symptoms of those conditions also transfer across, which is a sign that to a degree the microbes are not just going along for the ride, but they're also grabbing the wheel. Some studies have gone a little bit above and beyond these types of experiments and really sort of gone to town establishing a causal influence. And I wanted to just highlight one of my favorite ones from last year. This is by an Israeli group led by Aaron Segel and Aaron Elenov. It looked at a thing called the Yoyo Effect, which is the idea that if you diet, you might lose weight, but then you're very likely to put all that weight on and then some. So why is that? Well, first they established a mouse model. They feed mice high-fat food. They put on weight. Put them normal food. They lose weight. Give them high-fat food. They put on even more weight. So they also show this yo-yo effect. You look at their microbiomes and you see that when they start dieting, their microbes change, but they change with a time lag. There is a delay between their loss of weight and their microbiomes kind of normalizing. And that delay doesn't affect other factors that they looked at, either other physical traits or other things like lipids in the blood or blood sugar levels. It was just the microbes which were hanging back. So they did experiments. What happens if you remove those microbes with antibiotics? Well, the yo-yo effect is ameliorated, it's reduced. The mice are less likely to put on more weight when they start eating a fatty diet again. What happens if you take their microbes and put them in other rodents? Well, some of that bouncing back moves across as well. Can they identify a kind of mechanism behind it? Yeah. They looked at the ability of some of those lagging microbes to metabolize nutrients called flavonoids, and they showed that if they gave flavonoids to the rats that had dieted, they were a bit less likely to put on a lot of weight when they ate fatty foods again. And finally, they even used machine learning to look at the individual microbiomes of the dieting rats to see if they could look at their microbe communities and predict their likelihood of putting weight on when they ate fatty foods again. That's a lot of work in one paper, and I think it highlights the kind of clever experimental approaches that you can use to show that the microbiome really is having an influence on the health of these animals. But even in this case, there are still more questions and answers. Are the changes in the microbes instigating these problems, or are they just exacerbating them? What changes the microbiome itself in the first place? And to what extent do these effects that we see in rodents, in laboratory rodents, translate to humans going about in our sort of messy, realistic lives? And I think that's a critical issue for the field as it moves into the future, looking at things like effect sizes, at whether the statistically significant effects that the results that they're showing in studies like these translate to actually significant effects to us going about our daily lives. And I'll give you a couple of examples where I think that tension is important. There have been a lot of studies looking at whether antibiotics given to animals at a young age can increase their risk of weight gain and other conditions later on in life by disrupting their microbiomes. And a lot of those studies have found indeed that animals that get doses of antibiotics when they're young show changes in their microbiome that then correlate with a increased risk of obesity later on in life. Does that mean that taking antibiotics as kids makes us fat? Well, in these studies, a lot of the changes are actually quite small. I think in one of the classic ones, it was just like a 10% increase in body mass, which is like the equivalent of me putting on an extra like 15 pounds or so. That I probably don't want to, but it's not going to tip me over the edge into becoming obese. And likewise, we know through a lot of other studies that that antibiotics can change the microbiome, but that often these communities bounce back. So to what extent is this relevant to human health? I think it's unclear still. Here's another example. Much has been made about the effects of things like caesarean sections on the microbiome. The idea is that when human babies are born, we get our first doses of microbes from the mother's vaginal tract. And those microbes are our first colonizers, the pioneers of our own ecosystems. But if you're born through a C-section, those early colonists, rather than coming from mother's vagina, are more likely to come from her skin or the hospital environment. And could those early differences set babies up for changes in the way their communities then develop over time, leading them to poorer health? we know that if you look at the microbiomes of adults who are born via vaginal delivery or C-sections, you don't really see very many differences in their microbiomes, and any lasting differences. And there have been a few studies over the last year which have again found a lack of similar differences, even at babies of quite a young age. So I think the question is, those initial discrepancies in the microbiome normalize over time, but during the point when they are still separate, do those differences cause, do those increase the baby's risk of poor health later on in life? And how many of these kinds of disruptions do you need to actually increase the risk of disease? And I think we still don't know the answer to these questions. And I think we need to be very careful about communicating this type of work, because there is a tendency to sort of leap to conclusions in the field, to assume that changes in the microbiome are negative before we actually show that they are negative. So, you know, a lot of this is influenced by our priors, I think. A cesarean section feels like a strange, almost unnatural thing. And so we assume that a change in the microbiome as a result of this must be bad. Likewise, antibiotics feel like a kind of unnatural thing. So a change in the microbiome caused by antibiotics feels like it ought to be a negative thing, a dysbiosis. And dysbiosis gets treated almost in this weird sort of tautological way, where you know, a community is said to be dysbiotic because you find it in the body of a sick person, and they are sick because they have dysbiosis. So it becomes this kind of weird circular argument where dysbiosis becomes like the microbiology version of art or pornography, where you kind of know it if you see it. And I think we need to be careful here, because we are still at a very early stage of truly understanding the human microbiome and what it does for us, how it operates, what are the sort of guiding principles behind it. And we're at a stage where you know, studies are getting bigger, they're getting more powerful, and we're seeing that some early results aren't actually holding up. So there are classic results looking at obesity and the microbiome, showing that individuals who are obese, whether you're talking about humans or mice, have a higher ratio of one group of bacteria, the Firmicutes, to another, the Bacteroidetes. And this sort of FB ratio was a really important result in the early days of this field, but it turns out to be a bit of a mirage. So a lot of meta-analyses published over the last couple of years don't find consistent differences between the communities of obese and lean individuals. So you can see differences within particular studies, but none that sort of carry through across lots of different studies. Which means that I think it's hard to make clear pronouncements about the ways in which the microbiome is influencing our health. And we still need to be very careful about the way these type of results are conveyed to the public. So, and especially I think that's especially the case when we think about ways of manipulating the microbiome, which, you know, is sort of part of the reason why a lot of us are here, because we want to look at the science and think of ways of improving people's health. But here there is a lot of controversy too. So consider things like probiotics. These products supposedly contain beneficial bacteria that have all kinds of helpful effects for us. But if you look at large studies and reviews and meta-analyses, what you see is that probiotics are good for treating certain things like infectious diarrhea, but they otherwise tend to be a little medically underwhelming. They don't live up to a lot of the hype that surrounds these products. And I think that's because we're looking at the wrong microbes with these things. A lot of the strains and species that are in these products were chosen for historical reasons, because they're easy to grow and to manufacture, not because they are the A-listers of the gut or particularly well adapted to life inside us. A lot of them are industrialized proprietary strains, effectively domesticated microbes that have been grown in culture for a very long time. And they are found in these products in very low concentrations. A microbiologist named Mike Cox likes to compare these things to he compares swallowing probiotics to releasing a very small number of captive reared animals into the jungle and hoping that they will survive. And they don't, obviously. So perhaps rather than using this approach, it would make more sense to release large numbers of microbes that are very well suited to life inside the body. And that is certainly the logic behind a slightly unorthodox treatment called the fecal transplant. Everyone here heard of fecal transplants? Yes, good. They are exactly what they sound like. They are a way of remediating health problems by giving people an influx of stool from a donor. They are very difficult to find a good image for. Which is why I have prepared this technical demonstration involving emoji. So you take a sick person and you give them the stool of a healthy person and they get better. Was that clear? I can do it again. Okay, so this technique I think about like this as a way of returfing a lawn that has been overrun with weeds with fresh new pasture. And in this case, the weed is typically a bacterium called Clostridium difficile, or C. diff, that causes severe and very difficult to treat cases of diarrhea. Fecal transplants turn out to be one of the best ways, perhaps the single best way of treating C. diff infections. Antibiotic treatments cure maybe about a quarter of these cases of this condition, but fecal transplants have a success rate of upwards of 90% in various clinical trials. They are astonishingly effective. And as a result of that, people have tried these treatments for a wide variety of other conditions that have been linked to the microbiome, things like inflammatory bowel disease and irritable bowel syndrome and so on. But in all of those conditions, success rates have been lower and certainly much less consistent. Why? I think it's still unclear, because it is still unclear exactly why fecal transplants work. Indeed, several groups have tried to duplicate the success of these treatments by growing very specific cocktails of microbes found in stool and giving them in a sort of fake poop pill that takes out the ick factor from this treatment. But one of these attempts at creating a specific microbial cocktail, even though it went through very well in it succeeded quite well in early clinical trials, bombed out of a phase 2 recently, which allowed me to write this headline, which I should have really retired on that. I don't know why I carried on working. But even though this is very disappointing, I think it serves to highlight the fact that even in this case, when we are giving someone an entire community of microbes, it is still unclear how to do things like get that community to establish. We don't know exactly what it is about them that is conferring benefits, because, as I said, we are still at a very early stage in our understanding of this field, and because microbiome medicine is very different to what we're used to thinking of when we think about medicine. It is an act of ecosystem engineering. Unlike, say, imagine if you have a vitamin deficiency. You're trying to plug that by giving someone something, a simple act of addition. If you have a viral illness, you're trying to subtract, you're trying to remove viruses and get healthy. The math of that kind of medicine, oh dear God, I said math, I've been in America too long. The maths of that kind of treatment are very easy, but here, it's very complicated. It's like the difference between calculus and arithmetic. This is like shaping an entire world. Controlling something as complicated as a coral reef or a grassland or a rainforest with all the complexity that that entails. So no wonder this stuff is hard. No wonder it's challenging. I think there are a few options for short-term successes, like quicker wins in terms of manipulating the microbiome. One might be to not manipulate it at all, but to use it as a tool for stratification, to look at someone's microbiome and work out whether they should receive medicine X or Y. Because in a lot of different studies, we see that a person's microbiome influences their response to all kinds of medicines, things like acetaminophen, or the latest blockbuster cancer immunotherapy. So perhaps we can use a simple readout of our microbes as a way of directing treatments to the most appropriate patients. Maybe we could also drug the microbiome itself. There's a group that's done some really interesting work with heart disease. Chemicals in our diet, like choline and carnitine, get metabolized in our body to a succession of substances, including TMA and TMAO. The latter is problematic because it slows the breakdown of cholesterol and can increase the risk of heart disease, like atherosclerosis. So there are ways you could deal with this. You could deal with the enzymes that convert TMA to TMAO. Those are human enzymes, and you can indeed drug them, which knocks out the rest of that pathway, leaving you with TMA. Which sounds good, you have a lower risk of heart disease, but TMA happens to smell of rotting fish. So your arteries will be nice and clean, but you will never have sex again. So instead of targeting the host enzyme, maybe you should target this earlier step of the pathway when choline and carnitine get changed into TMA. And that bit of the pathway is catalyzed by bacterial enzymes. So if you knock those out, and there are indeed substances that might be able to do that, then you nix the whole thing in the bud before it can get started. And maybe this is a more suitable approach to reducing the risk of heart disease mediated by these kind of dietary nutrients. Okay, as I've tried to stress repeatedly through this talk, we are still at early days. We don't have yet a full list even of all the microbes that share our bodies. We can't really explain the vast majority of the variation between people. So my microbiome is very different to yours and yours and yours. But two of the largest studies in this field, published I think last year, tried to look at different factors that might explain their variation, and only managed to account for something like 8%, 10% of that variation in the microbiome across people. So despite looking at things like drugs, like antibiotics, diet, all sorts of other factors, they were largely in the dark as to why my microbiome is different to yours and yours and yours. When we look out into the broader world, the broader microbial world outside the human body, our ignorance, I think, becomes even deeper. So this image is a tree of life. This is a pictorial representation of everything that exists. And I want to draw your attention to the little green feather at the bottom. That little twig there represents us and every animal and plant and all the other life that you know and can see. The rest of this tree is microbial. And that purple spray on the right, representing a massive swathe of bacterial diversity, would not have existed on this diagram if I'd shown it to you about 2 or 3 years ago. So we are discovering corners of the microbial world at an incredible pace. Imagine what this diagram would look like if I showed it to you in 5 years' time, 10 years' time, 100 years' time. In the words of the wonderful Jonathan Eisen, this is humbling because we know virtually nothing right now about the biology of most of the tree of life. I Contain Multitudes is in part a call to explore that world. I think it is vitally important because this is the vast majority of life on Earth, and because exploring that world can sometimes provide incredible and unexpected benefits. And so the last story I'm going to tell you tonight is along those lines. It begins with this chap. His name is Simeon Bert Wolbach, and in the 1920s, he and one of his colleagues discovered a new bacterium that lives inside the cells of insects. And they didn't know what it was, they didn't know whether it was anything special, they ignored it for a long time. It took them 12 years to even give the thing a name, and they named it after him, which is why this bacterium today is called Wolbachia. Wolbachia is exceptionally common. This thing lives inside the cells of insects and in the cells of something like 40% of species of insects and other arthropods, which are themselves the most rich, diverse, numerous groups of animals on the planet. So Wolbachia can lay easy claim to being one of the most successful microbes in the world. And of course, these guys had no idea when they first discovered it. What does Wolbachia do? Well, lots of different things depending on which host it finds itself in. Wolbachia only passes down the female line from mother to offspring. And so males and sons are a dead end to it, and it has many ways of dealing with that. In this beautiful blue moon butterfly, in some populations, Wolbachia kills male embryos outright, so that the females outnumber the males by 100 to 1. In other animals like woodlice, Wolbachia transforms males into females as they grow up. In some wasps, it allows the females to reproduce asexually, cloning themselves, so they have no need for males at all. When it's not fighting the patriarchy, Wolbachia sometimes provides its hosts with a lot of different benefits. It does so for this bed bug. It provides it with vitamins that are missing from its diet of blood. It does so for the insect inside that green patch at the bottom of this leaf. That green patch is the work of a thing called the spotted tentiform leaf miner, a caterpillar that eats leaves. It uses Wolbachia to stop leaves from turning red in the autumn, giving it this little green haven in which to continually feed as the world turns to orange around it. So, Wolbachia is really cool, but the host that most interests us today is this one. This is an insect that does not naturally carry Wolbachia. It is Aedes aegypti, the mosquito that spreads diseases like dengue fever, yellow fever, chikungunya, and Zika. This thing, as I said, does not naturally carry Wolbachia, but for about 3 decades, a group of Australian scientists have been trying to get the two together, to insert Wolbachia into these mosquitoes. And they have succeeded now. Why? Because if Aedes aegypti carries Wolbachia, it becomes unable to spread the viruses that cause all those diseases that I told you about. It transforms from a vector into a dead end. And because Wolbachia is so good at manipulating the reproductive lives of its host, it spreads like wildfire through natural populations. So the idea is that if you release a small number of Wolbachia-infected Aedes aegypti into the wild, then very soon all of the local mosquitoes will carry that bacterium and be unable to transmit diseases to us, which would be a good thing. That approach has been trialled in various parts of the world, in some small pilot studies in Australia. This graph represents one of them. The grey bars show weeks between January and May of 2011, when those Wolbachia-infected mosquitoes were released. The green line represents the proportion of wild mosquitoes that carried the bacterium, rising from 0 to almost 100% within just a few months. So the approach works. Does it work for actually driving down the incidence of disease? A little cliffhanger while I drink. We're finding out because this approach is now being tested in large tropical megacities around the world, in places like Colombia and Brazil, Vietnam and Indonesia. And we should get some answers soon, but I think that there is tremendous potential in this approach. Unlike, say, the use of insecticides, which are toxic and need to be continuously resprayed, Wolbachia-containing mosquitoes are safe, and they only need to be deployed once. Once you do that, they are self-sustaining. And because it seems that Wolbachia prevents mosquitoes from spreading these diseases, from hosting these viruses effectively through many different mechanisms, it may prove to be harder for the viruses to evolve countermeasures, ways of resisting this approach. But really, I think this is a story about unexpected possibilities. Wolbach and his colleagues had no idea that any of this was possible. They in fact died, Wolbach in fact died in the 50s before anyone realized how common Wolbachia is, before he appreciated that his own name had been grafted onto one of the most successful pandemics in the history of life on Earth. Let alone that that organism could hold the key to treating, to fighting a lot of important illnesses that affect hundreds of millions of people. And that to me is the story of animal microbiomes in a nutshell. For the longest time, we have ignored microbes and we have feared them and we have treated them as enemies. Only recently have we truly begun to appreciate how important they are for our lives. Only recently have we begun to gain enough confidence to try and manipulate our partnerships with them. And even though our attempts to do so are still quite basic and a bit stumbling, there is huge potential here, and I think it's potential that stems from that basic curiosity to learn more about the world around us, that same urge that drove people like Leeuwenhoek to look at random bits of water under his microscope. And it's that urge to learn more about our world and the things that we share it with and the things that we share ourselves with that is what I hope to convey to people who read I Contain Multitudes. Thank you very much.
Well, I don't know about you, but I thought that was absolutely fantastic. Thank you, Ed. So what we're going to do now is I'm just going to take some questions from folks. We have a couple of microphones. Are we going to move them around? Is someone in the back? That's going to happen. Yes, we're going to move them around. If you'll point to people who raise their hand, a microphone will be taken to you, and you have an opportunity to ask a burning question about what Ed's talked about tonight, or I suppose any other issue in life that you might have.
Hello, there's one over there. Let's start with you.
Hello.
Hi.
I was wondering if you could leave us with some of the most actionable findings from the literature as far as the effect of dietary choices on the microbiome.
Okay, so I get this question a lot, and I usually don't love giving dietary advice, especially because I sort of feel that the more specific dietary advice gets, the more likely it is to be wrong. So I'm going to give you some very broad advice. I think that it is a reasonable thing to say that eating a lot of fiber is probably good for you and your microbiome. Fiber consists of a wide variety of carbohydrates that nourish an equally wide variety of microbes in your gut, and it seems to be a good way of feeding your multitudes that sort of, reproducibly found across a lot of different studies. So eat fiber. Anyone else?
Oh, there's no mic there.
Oh, right.
So I'm one of those people who spends lots of hours at the microscope, even though it has absolutely nothing to do with my day job.
Okay.
When I hear you talk about the Wolbachia thing, my alarm bells immediately go off, because I think about how many times we thought we got it, and we're going to eliminate this one thing that's going to take care of this other thing, and then there's this cascade because we don't understand the multitudes.
Yep. Of course. So this is a really good point. Is Wolbachia going to be like the new cane toad? Or, fortunately, the team that have been behind this are Australian, so they know well that risk. They have, I think this is actually, one of the reasons I'm quite fond of the Wolbachia story, and I tell it a lot, is that I think it's not just a story about interesting basic research having applications, but it's also a story of very careful science. So those guys went and did a huge amount of community engagement even before they had a mosquito with Wolbachia inside it. Like before their experiments worked, they were out in the communities, explained to them what they wanted to do and getting people's concerns. And many of them had similar concerns to what you're expressing now. So they did studies to address those concerns. They looked at things like, could Wolbachia get into other organisms that eat mosquitoes? Could it get into people who the mosquitoes bite? So they have thought this stuff through very carefully and very systematically. And I think as a result of that, they have huge levels of community engagement in the places where they're doing these pilot tests, which, when you think about it, is kind of an astonishing feat, right? Because you have to release these things on, at people's houses. So you're effectively saying, hi, you don't know us, we're scientists. We've loaded these mosquitoes up with a microbe you haven't heard of that's really good at spreading through wild populations. We'll need to release them on your property. These insects will probably bite you. We've never done this before. Are you up for it?
I'm going the total other direction. I'm thinking about the bats and the birds that ate the mosquitoes and what sort of traffic cascade might happen as a result of wiping out this one specific.
Aha, okay. So the wonderful thing about this technique is that no mosquitoes are harmed in the making of it. The Wolbachia approach doesn't kill mosquitoes. It just stops them from transmitting viruses.
Not from reproduction. Okay.
Correct.
Okay, all right. I withdraw my question. Thank you.
No, it's a good question. Hi.
So I'm reading the book, and it's fascinating to hear that you were speaking tonight because it addresses the question that one doctor has a lot of case studies, and without going into detail, he's saying that our microbiome directly influences our health, either to the benefit or not. A lot of the bad foods people eat can create bad microbiomes, and you can re-engineer your microbiomes by carefully studying your diet, looking at different foods that are more beneficial. Is that true?
I do think that there is a huge amount of potential there. I think that's hopefully where we're going to go in the future. But I think that you can only do that in a reliable way if you truly understand how these communities work. What governs the composition of the microbes? How do they change depending on what you eat, depending on your immune system, your genes, all of the above? And I think, how do the microbes within that community react to each other? I think we still don't understand a lot of those really fundamental questions. We can't look at someone, I can't sequence your microbiome and say, you're healthy, or you're going to get this condition or that one. It's still, it's been a very descriptive science. It's moving towards being a predictive science, but I think it's not still finding its feet in that regard. There are so many examples where people have claimed the potency of some kind of medical intervention based on their own experience, and you need large, well-powered, well-controlled studies to make these kind of claims. And I just don't quite think we're there yet. Anyone from that side? Hi.
So I have a question about standardization, storage, and reproducibility of fecal microbiomes.
Uh-huh.
So that's an issue for biological control, for example, making sure that you have the same quality and quantity and
Right.
Active component when you put something out to get reproducible results.
Yeah.
And so it seems to me that with a fecal microbiome transplant,
Yeah.
that there could be the difficulty, and can you make up a whole bunch of fecal transplant material and store it or something like that? Or are people working on that?
Yes, you can. So obviously it was a little bit ad hoc in the early days when this was still like a kind of weird medical oddity. But now I think that trials have shown effectiveness. People are starting to work towards that. So there are actually stool banks where donor stool has been collected and frozen, converted into like capsule form. So we are working towards more standardization, which I think will probably be important for regulators to be able to assess these therapies properly. And then, of course, a lot of people, as I said, are trying to work towards things that don't involve any stool at all, like specific cocktails of microbes that can be drawn up again and again according to the same standardized recipes. I think the challenge here is that it feels to me intuitively the case that you probably won't get a one-size-fits-all solution. It's not like there's like one person out there with like the magic stool that will cure everyone else. And that person is like hooked up to it. I don't know. What was I thinking? Yes, but so we might need a lot of different treatments. Maybe you need a kind of personalized cocktail of microbes for one person's specific condition. And I think that's the way things will go towards, but then how do you regulate something that isn't standardizable? I think that's kind of an interesting open question. Hi.
In your new job with Atlantic Monthly, you have an opportunity to deal with some very controversial issues related to this sort of thing.
Mm-hmm.
For example, the effects of vaccines on babies. Has your employer come to you and told you to avoid those kinds of things at the moment, or have you any ambitions to tackle them right away, or where do you come out on that?
Well, my employer has made no diktats to this effect. I cover science which I think is interesting and rigorous and important. And that includes a lot of microbiome work and work looking at the social and medical importance of vaccines, which have done a huge amount of public health good. Anyone else?
So are all these little microbes like equal players, or is there a hierarchy? Is there the strongest?
That's a good question.
Strongest survive, and these little guys?
Yeah, that's a really nice question. So think about the kind of dynamics you see in the natural world that you're familiar with. So predators, prey, cooperation, conflict. All of those examples exist in the microbial world too. So there are food webs, there are microbes that cooperatively nourish each other, there are predators that take out prey, there are microbes that parasitize other microbes. It is a very rich world. So I'm not sure if that exactly answers your question, but there are certainly food webs. People are actually actively studying predatory bacteria as a possible way of treating infections. Anyone on that side of the room? Hello.
Hi. So you talked a lot in your presentation about the animal and the human microbiome.
Mm-hmm.
I was wondering if you've been following the research going on in the plant microbiome, such as with fungal endophytes, and if there's anything you found particularly interesting or significant in reading about that sort of research.
Okay, so I'm going to throw my hands up and say that I am probably quite ill-informed about that, if only because the book, I had to draw a line somewhere in the book which was already super wide-ranging. So I know much less about plant microbiomes than I do about animal microbiomes, but I think a lot of the same issues that I've raised today apply there too. Clearly, microbes are important to the development and to the lives of plants. A lot of people are looking at using that in order to, say, boost yields of crops or to make plants more tolerant to different environmental challenges. But again, you know, we're talking about manipulating very complex worlds, so you know, it's challenging. But I think that a lot of the same themes and fundamentals apply across. You know, these are not organisms that are individuals in their own right. They exist in communities with other things around them. Anyone in the center bit? No. Okay. Oh, and the back.
Hello.
Hi.
As someone who's read a lot of literature of the microbiome, I was wondering how you approach writing so it appeals to a broader audience.
Okay, so, man, this is engaging a very different part of my brain. So I think it's really important with science writing to never underestimate your audience's intelligence, but to never overestimate their knowledge. And those are the two classic mistakes that one makes. So, you know, with something like microbes, you know, or with any field of science, you avoid jargon, tell stories, try and find things that will catch the interest of people who have not heard anything about this field before. And I want to emphasize the value of stories, because that is the approach I took in the book, that good science writing isn't just the act of translating papers. It is the act of revealing what it is like to carry out science. So, you know, in the book, I tell the stories about people who've worked in this field. I look at the way their lives, their thoughts, their ideas have changed over time. And I think that is a really important way of getting people to understand things and care about things that they might not necessarily care about. You know, we need to remember that when you're writing to people, to a sort of general audience, there's no particular reason they ought to be reading or paying attention to you. You need to earn that attention. You need to give them a reason to care. And I think when you talk about something like microbes, which are a little bit abstract, you know, most people have never seen these things. Most people are unfamiliar with them. It doesn't just work to say you need to care about this. And one easy way of making people care about microbes is to make people care about the people who care about microbes, which is why stories are a regular part of the book, which is why I went to and talked to a lot of scientists and went out to meet them and see their labs. Does that help? Hi.
Oh, yeah. I guess kind of extending on that and this gentleman's question, one aspect is writing to appeal to a large audience, but maybe there's the other aspect that's important is credibility, right, when you're communicating science. And I know I really appreciate it when you say like, okay, I don't know that, right? I just wonder if this is something you really actively think about, if you have the urge to explain more than you maybe should sometimes, or how do you feel like you gain the trust of your audience?
Yeah, I think that's really important, and actually it's sort of shaped a lot of my thinking about this field and how I cover it. The thing to remember is that so I'm a journalist, and as part of that, I'm not meant to be a cheerleader for science. You know, I'm not meant to, my work isn't necessarily a celebration of science. I'm also meant to act as a watchdog. You know, I am meant to look to point out flaws in studies as well as strengths. I'm meant to tell people what is not happening as well as what is, which is why, you know, in the book and in the talk today, I've been trying to be very clear about where things, for example, are hyped and exaggerated, where we don't know things as well as where we do. And I think a lot of science writing fails in that respect, that it's a little bit too rah-rah, go science, and not critical enough about the field that it covers. And especially in a field like this where you have a lot of fresh, nascent energy and funding and excitement, there is that temptation to go a little bit beyond what you've shown. And my job is to take a few steps back and to act as that voice of caution and restraint, which is what I try and do in the work. And I think that not only is important for my integrity as a journalist, but also for capturing what science is really, because it's not a series of discoveries and facts. It's a process of gradually reduced uncertainty, and I think this approach more reasonably conveys that. Hi.
This might be a complicated question.
Okay.
I've heard that there's doctors in New York, maybe Cornell, I can't remember the name, like Lagone, whatever, and basically they have a microbiome lab, and they will test your stools or whatever and see what types of what your ecology is, and see what's deficient and fix you based on that analysis. Has the have you looked into that to see whether it's successful or is it just placebo?
Okay, so I specifically chose not to take part in any microbiome sequencing as part of the research for the book, and I am still sort of skeptical about the personal value of stuff like that. So Jack, who's sitting just behind you, works in the American Gut Project, where you can get your microbiome sequenced, and I think there's value in that to sort of as an act of taking part in science, of contributing to research, but I don't think one should do it in the hope of getting answers back that are actionable and that will help you improve your health. Because like I said, I just don't think we're there yet. I don't think it's possible to look at someone's microbiome and accurately and responsibly tell them you are lacking in this, you need to do that. It's still a bit like reading tea leaves. So I personally would not do that. Okay. Anyone else? Hi.
Do you have any strong feelings about antibiotics?
Yeah, I do. I feel that antibiotics have been really important, and I think that there is a risk in talking about the microbiome to sort of unfairly demonize them. I think, we living in this age where a lot of infectious diseases have already been brought to heel, run the risk of being a little bit cavalier about how horrible a lot of these conditions were, and what it was like living in a pre-antibiotic era. So I think we need to recognize that they're important and that they are, running becoming obsolete because of the rise of drug-resistant microbes, and we need more of them. But I think we also are starting to appreciate that antibiotics are a very unsubtle weapon. They harm the microbes we rely upon as well as those that might cause us harm. So we need better ways of developing antibiotics or ways of treating infections that are subtler and that are more specifically targeted. Yeah, I think so. I think there is, I think the really important message with antibiotics is to not take them wantonly. So don't pressure your doctor if you have a cold, a viral disease that an antibiotic is clearly not going to be able to treat. Likewise, we need to massively reduce or cut out altogether the use of antibiotics in agriculture. Right, so there are a lot of things we need to, there are a lot of ways in which we can better use antibiotics more judiciously so that if we do actually need them, firstly, they're around, and also we've not already bombarded ourselves with loads of the stuff. Okay.
All right, well, I think, okay, I'll just shout. Well,
Or there's one right here.
Or here, have one of my two.
Well, I think with that, we'll adjourn for this evening and all go nourish our microbiomes, because it's getting a little late in the evening. But I want to, just before we leave, just thank Ed so very much for flying all the way out here. And